Now it's time to bring in our last panelist, Jerome, who gives us the view of another big feed producer, and he's a researcher, and so he's also deals with the development of new things, and he can tell us what he thinks about what he heard today.
Okay, thank you. First of all, thank you for the invitation. And yes, today, from my side, I will have the opportunity to present you the point of view from the industry. Yeah. The point of view from the industry and from industry that is producing feed additive for animal nutrition and health. So first of all, I would like to present to you the research topic of DSM Firmenich in terms of animal nutrition and health. So we are developing different categories of product, such as enzyme, carotenoids, vitamins, mycotoxin deactivators, or eubiotics. And these projects are actually trying to solve the different issues of the customers, such as reducing emissions or improving performance, and also, for example, helping to tackle antimicrobial resistance. So that's the reason why we also need to have in vitro models and in vitro tools to be able to develop feed additives and then to sell. Okay.
Is already off.
Yes. Okay. So yes, we need these in vitro models to be able to show the benefit of the feed additives for the animals. So I just summarized in actually in one slide the different topics I would like to discuss. So for DSM Firmenich, yes, this is a need to implement an in vitro strategy to develop new products and new solutions for the customers. So why do we need to implement in vitro models and in vitro assays? Of course, we already mentioned this aspect of the animal welfare and also the 3R principle, because we are facing more and more with ethical committee as well when we want to implement in vivo assay or in vivo trials. That we need to reduce the use of the animals, and we are also facing some question about do you have already showed some in vitro data about the potential of this feed additive? So this is a need, and also the implementation of in vitro assay can also reduce the cost and reduce the time when we want to develop feed additives. And it's also very useful when we have a high screening capacity, because if we want to screen a high quantity of products, for example, when I say high quantity of products, when we have our colleagues from the in silico department and coming, okay, we have 3,000 of candidates that could have potentially an impact of or benefit for the animal. Okay, maybe using animals and testing 3,000 of candidates, it will be a bit complicated. So that's the reason why we need to have a solution, a tool, to be able to screen this high quantity of candidates. Also, in vitro models are really great also to show the mode of action of this solution, of this product, to be able also to know our products and to show to the customer how the products are working to the animals, into the animals. So the second question is, what are we testing in our in vitro models? So for DSM Firmenich, the idea is to be able to show that our product, our solutions are improving the gut health, not only the gut health, but in this case, especially today, I would like to focus on the gut health. We also need to show that we are increasing the intestinal integrity, increasing the intestinal health of the animals. We need to show that we are improving the nutrient absorption of the animals. And also one thing that I think is very important is the host-gut microbiome interaction. Because the microbiome is part of the game, let's say. I'm not saying this because I'm a microbiologist by training, but I think that's really important to mention it in this case. Because yes, the gut microbiome is producing metabolites. And one topic of our research is also the gut-brain axis. And we are also trying to develop solutions that are modulating the microbiome and then modulating the metabolites produced to be able to have an impact on the welfare of the animals. And finally, another aspect is also the pathogen-cell interaction, for example, that we can also show in a simple in vitro assay. For example, when I speak about monogastrics, I know that, for example, E. coli in the intestine is quite an issue. So we are able also to show this direct interaction between the cell and the bacteria, or the adhesion, for example, to try to develop solutions that can inhibit the adhesion of this bacteria. So how can we implement this, or how can we use this in vitro platform? Of course, we need, and it has already been mentioned, we need to do compromise between the high throughput and the physiological relevance of these in vitro assays. So yes, as I mentioned, if you have plenty of candidates, we cannot use very complex in vitro models because we're not able to test all the compounds. It will take years to develop a product. But if we have simple in vitro assays, even if it's not completely physiological relevance, but we are able at least to narrow down the number of candidates and the number of molecules that we want to develop, it will help to speed up the project, and it will help to also to select the most potential candidates. But of course, we need to have also new approaches, more complex approaches to add in the panel of the in vitro assays that we have to, at the end, to select what are the best in vitro models at what stage of the project. At the beginning, to test simple in vitro models, and then to add more complexity when we have selected the final candidates. And also, that is one case that we apply sometimes in monogastrics, is that once we have finalized an in vitro screening process, we also have the opportunity to run small scale in vivo trial with a low number of animals to be able to have an intermediate solution to show that what we have screened, what we have showed in vitro has worked in vivo in a small scale trial before going to large scale in vivo trials. So all of these points that I just mentioned can maximize the potential of the in vitro platform for the in vivo success of developing a new solution for the customer. So what about aqua specifically? Because I was just speaking, yes, about aqua, not only aqua, but also monogastric poultry and swine. The species specificity, we already mentioned it before. So when my colleagues from the aqua group, so our scientist colleagues are asking me, for the customer, we want to show this and this. How can you do it? How can you do it? Okay, we have a human model, we have a piglet model, but I'm sorry, I have nothing to show with the aqua cell model. So that's technical limitation, unfortunately. And I think this is something really important for the industry to be able to show that the solutions that we are developing are based on solid in vitro aqua models. Also, regarding species diversity within the aqua, so that you already mentioned, that is also very important. And what I would like also to mention is the tempo between also the aqua academia and industry collaboration. So for the industry, we need to face also with the timelines of projects, and we also need to be flexible, but also to fit with the timeline of the project and to be able to say at one time, okay, it takes too much time. Unfortunately, we need to stop and we need to go on in another direction. Also, the in vitro models that are used for the development of solutions for the customer needs to be accepted also by the customers. Because we need also to show that, okay, what we are showing in vitro is valid in vivo and can show really the benefit that it shows in vitro. And finally, not only the acceptance of the customers, but also the acceptance by the regulatory bodies, of course, to be sure that, yes, what have been shown in vitro can be valid and can be extended, can be translated in the in vivo situation in aqua species. So that's it for my side. That's what I wanted to show you. Yeah, really the process of the in vitro development in the frame of the new solutions for the customer.
Thank you very much, Jerome. And thank you. And again, now the discussion is open. And if you want to ask questions to members of the consortium or to anybody else, if you are aspects that we haven't got time to explain this morning, we will be happy to extend this afternoon. And if there are other questions coming from the in silico attendees, maybe we can keep this up again so we remind ourselves. And Amira's.
Hi, so this is a question for you, Jerome. So you mentioned the in vitro model that you guys have for the monogastric. So can you, since we're in aqua, we like to follow the other animals. Can you tell us what took in order to establish those in vitro models to make the acceptance by the customers, which I assume are the producers of feed, and the regulatory? What were the things that you had to do?
So for the acceptance of the customer, the idea is really to show that we need to have, for example, cell line, or we need to work on microbiome that are coming from the species of interest, because that's one of the most important thing is to be able to show an effect of a product that has a benefit on cells that are coming from the species of interest, because this is the best way that we will be able to sell actually what we are developing, and also to be able to show that our in vitro models, our research is solid and that it's reliable.
If I can follow this, so can you give us an example of how a pipeline of such a development would look like in DSM? So
yes, so I can take the example of, for example, a product, a microbiome modulator, for example. I was speaking about the gut-brain axis, for example. So we need to show that a specific molecule is able to modulate the microbiome, and then modulate the metabolites that are produced that could have an impact on the host, on the pig, for example, in this case. So the idea is, how did we do? We just take microbiome from piglets, for example, that we isolate, let's say, and that we let in contact, or we let utilize the different products that we want to develop, and we then measure the microbiome modulation and the metabolites that are differentially produced and so on. And then these are put in contact with cell-based model with piglet cell line. And in this case, we really have the microbiome from the pig, we have the cell line from the pig, and then we can measure the different parameters that could be linked to the response of the metabolites produced by microbiome. And also we can measure pathways, for example, that are modulated in the epithelial cell line, for example, in response to the metabolites produced. And this is how we are trying to really to be as close as possible to the species of interest. Of course, the in vitro model is not ideal because we are not, this is just the discussions that we have had just before. We have not a complete model with all-in-one, but we need to have these separate steps and try to rebuild all in one and try to show that the different tests that we are applying could make sense.
Perhaps I can ask a question?
Sure.
Because it applies to these potential industrial applications. So I guess it's a question both to Jerome, but perhaps also to Francisco, who has the long history of having customers using these approaches, and perhaps also a question related to Pedro, and also with regards to who are potential customers and users of this. And you can decide who will answer, but perhaps to Jerome. I mean, this fish AI prototype that you now have been hearing about the whole day, is it of any use for you? And how do you see that?
I would say yes, of course, first. Because yes, as I mentioned just before, yeah, it's easy to work on human cell line. It's easy to work on piglet cell line. But yes, we are facing, let's say, lack of sharing of internal research in, for example, coming from different industries. And internally also, when I'm speaking about DSM Firmenich, we have scientists, we have great scientists, but regarding the timeline of the project, the time we have to develop a product, it's almost impossible to spend 100% of our time to try to develop a cell line that will respond specifically to the question that the customer had today, and it will be not the same tomorrow. And that's the reason why I think it's really important to have the possibility to have easier access to what is developed elsewhere, to be able to do applied science, to be able to develop new product, new solution, to speed up the projects, and also to speed up the applied research. So I don't know if somebody else.
Well, a lot of our results are already published, so this is a demonstration that those some of the contracts we have had with companies, for example, a French company was interested in comparing different commercial phytases using exactly the same platform. And they were interested in having this in a short time because they were the intermediates for another company. So they were only collecting the results, and they contacted us. we offered the platform, we offered the results, and they agreed that they were very interesting because they need this as an initial step to develop the in vivo trials, but they have preliminary information about the behavior or the results that may be obtained with the different phytaces, and they were very in agreement with our results. So there were 5 different products, and 2 of them were a disaster. So it's a completely worse. So this agreed very well with their preliminary information, and it's an example that we were able to develop and to obtain results using a simple platform in a short time, using, in this case, I remember it was a CBRIM. It was a CBRIM platform. So this is an example that this may work. And we have also examples. At this moment, we are currently running assays for another company that is testing bioactives coming from plant origin. We don't know which ones, but they want not only they're only interested in see and to test the effect of the digestion conditions on their product. So they initially they were not interested in including them in a matrix, but only to test them pure. But talking with them, we convinced them that maybe more interesting to use the matrix effect I mentioned before. And they say, okay, this is maybe more interesting for us. So which kind of matrix can you prepare? And we discuss it with them, which ingredients to select. Okay, we want those ingredients, put this in those dosages. So they selected, they say with dosage, and we simulated 2 different fish species. And now the assays are currently running. So I don't know which will be the results, but are examples of practical applications of those assays.
Do you wish to? No. Thank you.
Okay, then. Question over there.
I have a general question about fish AI and the simulation of the intestine. The factor that is somehow is neglected in this essays is that the role of intestine cells, because all the essays, as I understand, that's based on the concentration gradients that's exchanged in the membrane, and the role of cells to absorption, the active activity of them. Does it affect or cause the deviation in the final result?
Okay. Then I'm not sure I got exactly the question, but yeah, you're right. Basically, the result we all showed today has mostly to do with the integrity of the cell barrier, not to the activity of the cells themselves. And you're perfectly right on that. It would be a big limitation if we only had that. The thing is that we have also data dealing with amino acid absorption, and well, some of this has been also presented with the dipeptide absorption and activity and metabolism. So these assays have been performed. We haven't been able to show all the data, and we haven't been able so far. We hadn't got basically time to utilize all range of available assay in testing systematically the 5 dyes, because that is a big work which is still going on, and hopefully will go on even after the end of the project. But the tools are there in order to measure these activities, and now that we know how to handle the whole components, so the device, the cells, and in vitro digestion, now we are in the position to begin to move forward and indicate and study more in detail the absorption mechanisms themselves, which I fully agree that are very, very important. We show in some of our previous papers the enzymatic activity of the cells and how that is affected according to the exposure to different diets, but not in a systematic way as we tested the data that I show you today. So we had more isolated data because there came along different set of experiments. But definitely this thing is possible. And this brings me to, if there are no burning questions from the audience from remote, to raise another point, maybe so that we are close towards we are moving towards the end, because I see there something that we haven't talked much about it, that organoids are sort of on the sort of the top end of things. And as Nicole mentioned today, we have been facing with this sort of choice and sometimes criticism that there is a widespread impression that organoid is the top of the system, which we were not, which we did not fully agree with that, especially in the context of functional tests, as we were mentioning now. Because in order to do this functional test, you need an easy access to the lumen, and then you have to have a clearly separated session where you can analyze what comes through the barrier. And organoids cannot provide that. The classical organoid is a clump of cells which is embedded in a collagen matrix. Most in general, a matrigel matrix, which is not even a collagen, is a basal membrane, so it's not physiological at all. And so there is no way in which you can administer the IVD to the right side of cells. So organoids are useful for many, many things. And they're organoids of all organs. So we are just discussing about the enteroids, so the organoids of intestine. So maybe are useful for cancer studies because they are looking at different interactions. But I'm not so sure they are appropriate for absorption of functional studies of the intestinal lumen. And I don't know if somebody want to, well, the idea is to stimulate a bit of discussion. So I'm pushing this a little bit. So both here in the room or from the audience from remote. So I wouldn't put in any way organoids as top choice when we are dealing in this kind of functional test of the intestinal wall. For our purposes, or even in other species like human or other domestic species, if you want to study absorption and digestion and nutrition, they might be more suitable for pathological models. That's what I would throw to the audience and see if somebody agrees or disagrees.
You may think about that. While you are thinking, there is a specific question to Jerome for I guess from the academic side to an industry side. What are the process of clearing a new ingredients, such as a new source of protein? What are the regulatory hurdles? How to overcome it, and how long time does it take?
Take us to the very complex.
Yes, that's a very complex question. I don't know if I could answer that question. But if I can talk about feed additives specifically, not about single ingredients, but feed additives. Yes, we are facing issues regarding, yes, regulatory, IP, also toxicity evaluations, and that's the things that are taking quite a long time when we are developing new products. So I don't know if this is what the person who asked the question is expecting, but yeah, and regarding the timeline, it's difficult for me to answer that question. But it's taken into account when we are developing a product. We have these different phases of the development of a product, and this is generally at the second or third part of the product after the in vitro screening part that we are then facing to this or regulatories or IP topics.
Is there a comment or question related to?
General comments. So I can be last.
No, okay. General comments be last, okay. But maybe before, if there are general, maybe we can leave that and follow up on these organoids, or because you were asking, I guess, more of a statement than a question. But access to apical and basolateral compartments is fundamental for the intended use. Organoids are not. Can I repeat the statement? It's more of a statement than a question. Access to both the apical and the basolateral compartments is a fundament for the intended use that we are talking about here today, whereas organoids are not.
Yeah, so thank you for the support.
Would you agree? Or someone disagrees? Please.
Yeah, so
Okay. I guess I was the one that got the invitation to join the project, and luckily we had Trond that wanted to participate. I have to admit I was very skeptical when it comes to range, what kind of ingredients with an in vitro method. But I have been very lucky to follow the project, and I'm very happy with the result as well. However, I'm still skeptical towards in vitro methods. And I think they are useful if you, for example, for soy, which I have been working on all my life, at least my scientific life. If you compare the soybean different batches or processes, etc., I think an in vitro method can be very useful. But if you compare different feed ingredients, then I'm more skeptical. And I think we have seen this today as well, like the feather meal, which is not really fitting in as well in the different method, depending on the biomarkers you are using. And it's because an in vitro method is so static. You talked about how to make a standard mixture of enzymes that you can use so that you can reproduce but the gut is not static. It's so dynamic. It's amazing. I've been so surprised several times during my career that how it's working. And it started when I was a student and had to see what happens if I introduce protease inhibitors into my gut. What happens to the enzyme production? And at first, you can eliminate the protease activity or the immediately, but then after some time, a new trypsin, a new chymotrypsin appears. How this mechanism is working, we don't know. But after a while, well, the other proteins than the protease inhibitors are digested in a way, but it comes with a cost. But it tells how dynamic this is. So maybe it's because, I'm sorry, it's a little lecture. The feather meal, which is so difficult to digest because of all the cysteine bridges, which I guess the products are hydrolyzed somehow, but still a lot of sulfuric amino acids and many bridges that are not broken. But in the animal, I think we had some very good growth results for the feather meal. And the animal, the fish, has overcome this somehow. And I think we need to take into account the dynamics, and we can end up with some wrong results, conclusions, if we don't consider this and kind of set some limits for how to use in vitro methods. So, yeah, and another thing is that if you fast a fish, well, the surface of the mucosa will diminish to the half in a day or two. So that also illustrates the dynamics of the. And as we have seen, if you feed some rainbow trout in a breeding program a diet containing soybean meal, which they react with inflammation. But after 3 generations, the inflammation is gone. They can tolerate it. So it's a dynamic that is so enormous, and you can hardly find other tissues in an animal body that it reacts, responds, and adapts. And I think that's why we are not carnivorous anymore. We used to be some 1,000 years ago, but today we can tolerate the plants because there is a breeding pressure according to the diets you're eating. Okay, but I think I have been so happy listening today because we can learn a lot. And the 2 new cell lines, I'm sure we can use them as tools for future studies that can tell us more about the dynamics. And we can learn more about the intestines of the fish, which are so different and so interesting and so necessary to know. So, okay, thank you.
Thank you, Åshild. And yeah, please, yeah, sure.
Yes.
The pH in the salmon.
Please repeat in the microphone.
Sorry. You claimed that the pH in the salmon stomach was low. It's not. No, we have measured several times, and it's like 4.5 during a digestive process. Fasting salmon has a very low pH in the stomach.
So this confirms the need to have a physiological basis to develop the in vitro models. I completely agree with you, Åshild, because the in vitro simulations are only tools to help you to reduce the number of assays. If you make this preliminary screening, you can reduce, and focus only on some of the ingredients or some of the tests, so to reduce the number of fish. And also, it makes us, they make us to know more about the physiological interactions that we have. As an example, we have learned, for example, testing some companies, they produce enzymes, exogenous enzymes, the effect of exogenous enzymes. Okay, if you put exogenous enzymes in the feed, it's expected you can have a higher digestibility of the ingredients, let's say carbohydrates or let's say protein. Well, the proteases, exogenous proteases interact with endogenous proteases. So endogenous proteases, we have found that they cut and they destroy, and we demonstrated this and we learned this using an in vitro assay. So sometimes the in vitro tools may be useful to understand things that are more difficult to assess using in vivo trials. But of course, they have their limitations. But in the same way that they have limitation when applied to humans or to terrestrial animals, and they have been used for decades in humans and in terrestrial animals, and they have proved to be useful. So I think that the point is to be able to develop suitable models adapted to different targets and to different species, with the ability to have the physiological conditions in mind to develop those models. And it's only a tool that maybe have a potential that still is not explored at least in aquaculture. So for the future, we need to have the same kind of models that we have in the other species. It is okay for us, I think. Perhaps Fulvio agree with me.
As we said, whatever you look at it is very complicated, and if I knew at the beginning how complicated it was, maybe I wouldn't dare. And this is what I would like to mention, closing it, to mention the very origin of this project. This goes back to 2016 in the Giardini Naxos, where we were taking part to a cost conference that Tiziana was chairing then, and there was Amos Standler with us. I don't know if he's still online, so it's a special thanks to Amos, who really was encouraging me to actually throw myself in this adventure, even if I never saw a live fish before in my life. I usually saw fish only in the dish, but the proper dish. So I didn't know anything about aquaculture. but he was very positive and encouraged me to go through this. And after the first attempt that didn't work, it wasn't successful. We tried a second time and it was successful. And here we are here at the end of the project many years later, and well, much wiser. And we know how the whole thing is difficult, but still we got some interesting results, and hopefully they will be keeping on growing and keeping on developing. And so I think we have run to an end of the day, and Trond close the event now.
Well, I didn't know that Amos was involved in this. So hello, Amos, wherever you are. It's a pity that you are not here. Anyhow, I just want to stress one thing, to congratulate you on the project. And one word that comes to my mind is interdisciplinarity. Because here you saw a lot of experts from different fields that are crucial to have this model running. Because you need people understanding chemistry, gut integrity, and cell structure. So anatomy normally is not a very interesting field, and we need this kind of people like exploring what's the morphological aspects of the cells, people from the bioengineering and more engineering part that know nothing about the fish and don't really need to know, but they need to understand that biologists cannot cope with very complex devices, so simplicity is really something, because if you need the experts to run things, it will add another layer of complexity. So, and the need that different people from different fields get together to understand the microbiome and what's the impact on that, and to put them in the same room talking so we can move faster. And sometimes you need all these people bringing all these inputs. So I congratulate you all because of the different aspects that you managed to bring to the project.
Thank you very much. You're speaking my words. It's exactly what I was going to say, because I think also, but then we are on the same level. It's to me also, I think science is curiosity and collaboration is two very important factors. And hopefully this has been a good demonstration of both. So, and clearly we see that we are at the start, not at the end. So I think we should all learn from what we have been through today and take it out into our different activities, whether it's teaching or research or industrial application or other type of networking or dissemination activities. So with this, we will close the official part of the program. I would like to thank everyone contributing. First of all, to the panelists. We have a small token of appreciation for you, a reminding from NMBU. So please, to the external. I would also like to thank all the speakers today, and of course everyone attending the audience and making this a very nice and good discussion. That for the online attendees will now close. Thank you very much. See you next time. For those of you present in the room, now the fun begins, because now in about 7 minutes, there are drinks and more food for us available up in Vitenparken, which is the locals know where it is. It's about 5 minutes walk, but so we will sum up here, just gather our clothes and things and walk directly up there, and it will be the social part of the program. Very good. Thank you all.